All right. Welcome, everybody. This is a re-recording of the lecture that we went over in class. The original recording had some audio issues. So in this lecture, we're talking about fermentation and anaerobic respiration. All right. So if we're talking about anaerobic organisms, we're talking about organisms that don't need oxygen. All right. Some of them may never need oxygen, and they would simply be an anaerobic organism. Some of them can use oxygen, but when oxygen is not there, they can also survive on other means of respiration. And these would be called facultative anaerobes, right? They use aerobic respiration when there is oxygen, and cellular respiration can proceed as normal, but they can also survive on fermentation. These are typically going to be really small organisms. All right, so some organisms that might be anaerobic or facultative anaerobics, all right, could be bacteria, species of archaea, some fungi, and even our own muscle cells. So our muscle cells, when we are working out and we're not supplying enough oxygen fast enough to those cells, they can actually temporarily revert to fermentation. All right, so we're going to talk about anaerobic respiration and fermentation as two means by which anaerobic organisms can still get energy. So anaerobic respiration is a form of respiration that's very, very similar to cellular respiration that we've already covered. But instead of using oxygen as the final electron acceptor, it uses a different molecule. And we'll go over this a little bit more in depth. The other way that these organisms can get energy is through fermentation, right? And we've pretty much learned fermentation already, right? Fermentation, it's producing ATP in the absence of oxygen. It's going through the glycolysis cycle, which is also that first step in aerobic cellular respiration. But then we have this additional step of regenerating our NAD+ molecule. All right, so let's take a step back and let's look at aerobic respiration, right? This is cellular respiration as we have already learned, but I don't think I did a great job in explaining exactly how the electron transport chain worked. So the electron transport chain, as we see here, consists of different protein complexes that are embedded within the mitochondrial membrane. Now, in the electron transport chain, we are using our NADH molecules. We are also using our FADH molecules. Now, we're not using those as much, all right? The primary electron carrier here is NADH, and in fact, this image completely ignores FADH2 because it's not as significant of a player as NADH. Now, NADH, right? We know it carries electrons, and when it donates its electrons, it's also going to lose some of its hydrogen ions. Now, a hydrogen ion, right, so a hydrogen molecule is one proton, one neutron, and one electron. If a hydrogen molecule loses an electron, we would call it a proton. All right, so this H+ ion is also called a proton because it has a singular proton. So NADH, right, is donating its electrons and it's losing some protons. Now the electron is going to follow this path in yellow here, right? So the electron is going to move through these different proteins embedded in the membrane, and the movement of that electron provides energy for these protein complexes to move our hydrogen items from the mitochondrial matrix outside into the intermembrane space. All right, so it's the movement of the electrons that provides the energy to move the hydrogens across our gradient here. All right, now this process will eventually build up a large gradient of hydrogen ions or protons, and it's these protons that will then go through ATP synthase and create ATP. But now if we focus back on this electron, right, this electron that has been moved through, right, eventually that electron needs to be captured by a molecule, right? We don't just want a free-floating electron. It can't move on forever. It has to be captured by a molecule. And the molecule or the atom that captures this is oxygen, right? So oxygen is going to combine with a couple of hydrogen ions here and form water. So oxygen is the final electron acceptor. Once electron is done moving through the membrane, right, it's going to connect with oxygen, and oxygen will become water along with some hydrogens, and that is our final electron acceptor. All right, so that's aerobic respiration. That's when we have oxygen, right? But in anaerobic respiration, these are bacteria and archaea that can use other electron acceptors, right? So if they don't have oxygen available, they can use other molecules such as nitrate and sulfate. They can also initially break down different molecules besides glucose. So they can break down these other molecules that I am showing you here, and it's these molecules that can provide electrons that the NADH will eventually accept, bring to the electron transport chain, and donate it in order to make ATP eventually. All right, so we're still using the same steps. We're still using the electron transport chain. We are still using ATP synthase in our mitochondrial membrane. So let's walk through an example here. And so this is an example of a bacteria called E. coli. You might be familiar with this bacteria. And E. coli can go through anaerobic respiration. And instead of using oxygen as its final electron acceptor, it uses nitrate. And so if we look at the image here, it's going to look very similar to what you've already seen, right? NADH is donating these electrons. This electron is moving according to this red path here, right? NADH is also giving away some protons, and those protons are moving across the membrane according to this electron movement. We are building up a gradient here, and that gradient is what supplies ATP synthase with the energy in order to phosphorylate ADP, add on a phosphate group, into ATP, all right? Same exact processes. The only thing that is different here is instead of oxygen accepting that electron and becoming water, now we have nitrate accepting that electron, and that nitrate is going to become nitrite, nitrite, all right? They sound very similar, but they are slightly different. So our nitrate molecule accepting the electron will become nitrite. Everything else is basically the same. All right. So now let's talk about fermentation. So in fermentation, right, some organisms, they can only use oxygen as the final electron acceptor. So when oxygen isn't available, they cannot use other molecules like nitrate or sulfate, right? Instead, they have to go through the process of fermentation. All right, now fermentation is really a relatively simple compared to cellular respiration. There's fewer steps. So the two steps in fermentation is glycolysis, which we see in our image here. Right? We are breaking down a glucose molecule into two pyruvate molecules. We are making a small amount of ATP, and we are also creating NADH molecules. Now that next step in fermentation, and this can vary, right? But this next step has to regenerate our NAD plus molecule. So we have to bring NADH back into an NAD plus form. All right, and this is where the problem with fermentation lies, all right? So if we just did glycolysis, right, we would be producing NADH molecules like crazy until we ran out of NAD plus molecules and no longer could create NADH, all right? But NADH is a good example of too much of a good thing can be bad, all right? So while, yes, the cells use NADH, right? high concentrations of NADH is going to cause these NADH molecules to haphazardly donate its electrons to other molecules, instead of, say, going through the electron transport chain, where that electron is very much controlled in that it's going to be donated to oxygen or nitrate or sulfate. Right? So when it doesn't have that option, it's going to haphazardly donate its electrons. And when it does this, it can promote the formation of what we call free radicals. These are molecules that are just highly reactive. They can damage DNA and they can damage cellular proteins. Right? So they are very damaging molecules. We don't want to create these if we can't help it. The other problem with fermentation, I alluded to earlier, is that glycolysis uses NAD+. Right? And once we run out of NAD+, we can't do glycolysis. So we need a way to keep regenerating NAD+, to keep glycolysis running, to keep producing those small amounts of ATP. Now there's two ways that anaerobic organisms that undergo fermentation can do this, right? So in order to regenerate NAD+ and decrease the buildup of NADH, cells can either go through lactic acid fermentation or alcohol fermentation. We're going to talk about lactic acid fermentation first. And so in lactic acid fermentation, glycolysis is still the first step. All right? Glycolysis is happening as usual. But after glycolysis, when we have made these 2-pyruvate molecules, these 2-pyruvate molecules will be converted into lactate, right? And the process of converting pyruvate into lactate will regenerate NAD+, right? So it uses NADH, and once it's done using NADH, NADH becomes NAD+. And this NAD+ can then be used back in glycolysis. So that's one example of how cells can reduce the buildup of NADH. And a good example of this is in our muscle cells. All right, so our muscle cells will go through lactic acid fermentation. And once our muscle cells, say we're done exercising, right, adequate oxygen is once again provided to our muscle cells, that lactate can be converted back to pyruvate, right? Or that lactate might be used by the liver and other tissues to make glucose. And then glucose can be used in cellular respiration once again. Another good example of an organism that uses lactic acid formation and that we take advantage of is this bacteria Streptococcus lactis, right? As well as other types of bacteria that undergo lactic acid fermentation. But we harnessed what they produce in order to make cheese, right? So I like cheese. You might like cheese. We have these bacteria to thank for that. The other type of fermentation, right, is alcohol fermentation. So instead of producing lactate, now we are producing an alcohol, specifically ethanol. All right. So again, we're going through the glycolysis step. Glycolysis step has not changed. Once we have our 2-pyruvate molecules, we are converting those 2-pyruvate molecules into ethanol. All right. Now, a byproduct of alcohol fermentation is that some CO2 is going to be produced. So if you've done lab this week or will be doing lab this week, we are measuring that byproduct when we look at yeast alcohol fermentation. Okay. But this process, just like in lactic acid formation, also regenerates our NAD+ molecule. Right? So it uses up NADH, regenerates it to NAD+, and that NAD+ can then be used in glycolysis once again. And again, we take advantage of this and we use organisms that undergo alcohol fermentation, most commonly yeast, to ferment various types of alcohols. All right, so as a review here, all right, so fermentation, we are going through glycolysis, right? However, if we just stayed with glycolysis, we would get too much NADH and we would not regenerate NAD+. All right, and so organisms have evolved two different pathways to either make lactate or ethanol in order to regenerate our NAD+ molecule. And this image to the side here are just different ways of showing you that same exact process that we talked about in the last two slides. It's just drawn a little differently. All right. Now the big thing to remember here is that during fermentation, glycolysis is the only thing producing ATP. So for one round of glycolysis, we are only getting 2 ATP in fermentation. Versus aerobic respiration, when we have oxygen available, we can get up to 30 to 34 ATP. So there's a big difference here, and this is why we see this type of energy conversion in very small organisms. Organisms that don't have a large energy demand, such as yeast and bacteria. All right, so in class we did go over this case study in fermentation here, and so I will read this in its entirety here. So a 61-year-old man with a history of home brewing stumbled into a Texas emergency room complaining of dizziness. Nurses ran a breathalyzer test, and sure enough, the man's alcohol concentration was a whopping 0.37%, or almost 5 times the legal limit for driving in Texas. There was just one hitch. The man said he hadn't touched a drop of alcohol that day. He would get drunk out of the blue on a Sunday morning after being at church, or really just any time, says Barbara Cordell, the Dean of Nursing at Panola College in Carthage, Texas. His wife was so dismayed that she even bought a breathalyzer. Other medical professionals chalked up the man's problem to closet drinking. But Cordell and Dr. Justin McCarthy, a gastroenterologist in Lubbock, wanted to figure out what was really going on. So the team searched the man's belongings for liquor and then isolated him in a hospital room for 24 hours. Throughout the day, he ate carbohydrate-rich foods, and the doctors periodically checked his blood for alcohol. At one point, it rose to 0.12%. All right, so even after being isolated, this man still had a relatively high blood alcohol concentration, especially since he hadn't drank any alcohol. And so I would encourage you to pause the video here and answer these questions on your own to the best of your ability. So I will give you a chance to pause it, answer these questions, and then come back and we will discuss them. All right, so hopefully you had a chance to think about this on your own. So in our home brewing case, what is going on, right? This man is getting drunk without having drank any alcohol. Well, we know this man has a history of home brewing, all right? And this is really the key here. So in home brewing, right, we're using yeast to create alcohols. Yeast are very, very small organisms, right? It's likely that somehow he ingested some of these live organisms, these live yeast cells, by mistake, right? And then these yeast cells are undergoing aerobic respiration. They are oh my goodness, facultative anaerobes. Ooh, mouthful this morning. So they can switch to fermentation, all right? And when put into an environment like this man's digestive system, where there is no oxygen, it will produce alcohol, all right? So alcohol is being produced in the process of alcohol fermentation. That alcohol is then entering this man's bloodstream and getting him drunk, right? Now, eventually it seems like this yeast will eventually die off and this man will go back to not being drunk, right? But if he continues to home brew, he's going to continue inhaling these organisms and this problem will persist. So how can this organism live on such small amounts of ATP, right? How can yeast live on such small amounts of ATP? Well, yeast are very small organisms, all right? They don't need a lot of energy to sustain them. All right. Now in class, we went through this content check, all right? And For these content checks, the in-class portions are meant to be provided to you in class, right? And if you miss class that day, you are kind of out of luck, right? But I will make some exceptions, right? So if you email me and talk to me about it, I will provide you with the content checks, right? Likewise, if you just talk to me anyways, right, I will give you a chance to do them. I am pretty lenient here, but it's not something I'm going to go through the effort of providing everybody who misses class on that day, right? That's just one of the benefits of being in class, is that you can get the in-class portion of these content checks. So these next few slides I will let you go through on your own. You can kind of think about them on your own, but the questions associated with these different parts of our content check are not provided, right? So you will have to reach out to me if you need these.